All right, so the program that we're going to use to analyze your DNA sequences for your PCR project, and then we're going to use it again at the end of the semester to analyze your whole genome sequencing data, is called Benchling. It's free, it's online. So the first step will be to make an account. So you'll just click this link right here. It's going to take you to the sign up page. I just did this really quickly. I use my personal Gmail account, but I recommend you use your Wesleyan account, but you can do whatever you want. And clicked sign up with Google, and then it was super easy. When you get in, it's going to look like this. So the first question it's asking me is like, what kind of work do I do? So I'm going to choose academia, not industry, because we're using this for school. Where are we? We're going to choose Virginia Wesleyan. It says college because it hasn't been updated. You guys can choose undergrad. I'm going to choose professor. I just made a new account so that mine would look just like yours. And then what field are we in? Let's pick genetics and genomics. You can also just skip all this if you don't want to do it right now. All right. It's finding me. This is my research lab. So if you'll ever do research with me, you will continue to use your Benchling account. But we're going to not join a lab. Okay, so what do you want to see first? We are not going to use the lab notebook. We're going to use everything for molecular biology. So we're going to say, let's take me there. And one of the things that I think is slightly annoying about Benchling is that they auto-load some DNA sequences in here that you aren't going to use. And so they kind of get in the way. You can delete them all if you would like. All right, so this is a split screen view, which isn't my favorite. If you want to turn that off down here in the bottom right corner where it says split workspace, you can just click that and it will no longer split and it'll show you one thing at a time. So the tab you're in is which sequence you're looking at, and then you can see all these different things about the sequence. So sequence map, linear map, plasmid, if it's, you want to see where things are at. But for the most part, we're just going to be looking at the sequence itself. This one's got some stuff annotated on it. So these are restriction enzyme cut sites. We're not going to be using those. So if you want to make those disappear, over here on the right side where it shows the little scissors, it's asking you which ones you want to see, and you can just click none for cut sites visible on the map. And then all those will disappear. So it's a little less cumbersome to look at. And then this has a gene annotated for you with these boxes that are colored, and it has the name of the gene there. And then it also has the protein sequence on here as a translation. And what's cool about this is you can click on each one and the codons will appear. So that will be handy dandy later. And you guys go and figure out what your mutations are and what they mean. All right, but what we're going to do, we're going to start with making a folder for all your stuff to go in so it doesn't get mixed up with all this junk that they automatically give you. So I'm going to hit the plus sign here and I'm going to create a folder, and I'm just going to call it Bio 288. And then where do I want it? Example project is fine because that's the only project we have for now. All right, so now I've got a folder. We can't see all that other junk that we're not using. We're just going to have the stuff that we want. So if we hop back over to Blackboard, the things we want to add are the DNA sequences and the primers. So the DNA sequences are here. So you can open this link to this nice little Google Doc. And here is our E. coli 16S rRNA sequence. So I have already found this for you. It's ready to go. All right, so let's pop back over to Benchling and we're going to create a sequence. So hitting that plus again. Now I'm going to come down to DNA/RNA sequence, and I'm going to choose new DNA RNA sequence. A Benchling tip is when you're on a menu, you just have to hover and then the sub menu will appear. If you click on a menu where you were supposed to wait for a sub menu to appear and then choose something, it just disappears. So make sure you hover before you click on stuff, because that clicking this doesn't do anything. We got to come over here and click this one, new sequence. All right, what are we going to call it? E. coli 16S RNA. It's a DNA sequence. It is not circular, it is linear, so it's a straight line. We want it in our Bio 288 folder. Schema, we're going to ignore. And I'm just going to go ahead and click create. And it's going to create it blank. So it says type or paste bases here. So now I'm going to pop back over here, highlight this, copy, paste. And then it's like, you're about to paste a sequence. Are you sure? You have to hit enter. It's good they ask you if you're sure before you accidentally change your sequence. So, all right, so here's our sequence. It's got no annotations or anything. It's just very nice and plain looking. So we want to repeat that process with our A-bomb and our staff. So I'm going to pause my recording for a minute and do that. And then I'll be back. All right, I'm back. I've got all 3 of my DNA sequences in here now. So these are all here. Sorry, that one is in lowercase letters. Hopefully that doesn't drive anybody crazy. All right, so next step is we're going to import all of our primers. So we're going to go to that same plus sign. And this time we're going to choose oligo. Oligo, why do we use that word? It usually means like a short custom piece of single-stranded DNA, which is exactly what a primer is. And again, we're going to wait, hover on the menu, and then choose new DNA RNA oligo. So same thing, we need a name. It's going to keep it on DNA, keep it in the same folder, ignore the schema, and then put the bases here. So we're just going to pull these, oops, wrong tab, right here out of Blackboard. So these names, CWP stands for Caitlin Williams Primer. Sorry, that's kind of boring. That's just what I name all the primers in my lab to help keep track of them. And then here's that sequence. We're just going to copy and paste that right there. And then you can change the color that it shows up if you would like to. I tend to choose green for forward primers and then like a red or orange for a reverse primer. So you actually have one forward, which is this 23, and then all the rest are reverse. So I'm going to choose to make this one greenish, kind of like go as start, you know, and then we'll make all the other ones orange. And here it is, super easy. And it will open a tab for the primer, but you don't need the primer tab open. So I'm just going to go ahead and close that tab, because if I do that, I'm going to have too many tabs. And I'm going to go ahead and close this random plasmid that they started me with too. All right, so let's do that one more time, and then I will stop and you can finish the rest. So plus oligo, new DNA RNA oligo. We need a name, so it'll be 24. Get rid of that. CWP 24, and then here's our sequence. Copy. Whoa. Paste, and then again, this is reverse, so I'm going to leave it on this orangey color. Create. So now it should show up over here in my list. It does. It puts these alphabetical automatically, so that's why they're inserting kind of here in the center. And it makes the tab, which again, I'm going to get rid of. All right, so keep doing that until you have them all, and I will see you in the next video.